So welcome everyone to this ZOOMinar. Today we have two invited speakers from France. The first one is Rezaei, and the second one will be Sven Saupe. So we will start with Human. Human is a research director in the INRA, which is it's a French institute, a national institute of research for agronomy. Human got his PhD in 2001 in biochemistry and biophysics. And then directly after his PhD, he was hired by the INRA, and he started a group on prion biology in 2005. And since 2011, he's head of the group macro assembly and prion pathology. And he's going to talk about about protein misfolding and aggregation. So please, Human.
All right, thank you very much. Thank you very much to Lucy and to Hamstra to invite me to make a presentation, to present our works in this webinar. So I'm gonna share my screen first, and normally it should work. Sorry. I did, so normally it should work. Is it the right screen? I don't think.
Yes. No? Yeah. Oh. Well, we saw a presentation. I don't know if it was a good one, but we saw one.
Okay. Is it this?
Yes.
It's good. Okay.
It's perfect.
So,
yeah.
Okay. Thank you very much. So my talk gonna focus on prion pathology and the fact that in prion pathology, so prions are not simply a one given amyloid assemblies. There is multiple objects that coexist in the same environment. So my talk will focus on the dynamic of these assemblies and what's the consequence in prion replications, prion adaptation, and prion evolutions. So first, a few words about the prion pathology. So prion pathology is a neurodegenerative disease, is induced motor and sensitive dysfunctions, neuron loss, spongios, and astrocytosis. And I don't know if I show in the right screen, but here you have some illustration of spongiosis, normal and prion disease brain. Neuron loss and astrocytosis, and usually the evolution of always the evolution of the pathology is associated to accumulation of a misfolded PRP that's called PRP-SC aggregate of PRP. So you have usually a long asymptomatic incubation period. The issue is fatal. It means that once you start to have prions, you have to give up all hope. You're gonna die. It's mandatory. Without any conventional immune and inflammatory reactions, it means that you go to hospital, you can do whatever examinations. You cannot know if you have prion disease or not. And as I told you, you have an absence of serological symptoms, and all prion pathologies are transmissible. And as I said, that the prion pathology is a consequence of transformation of a normal healthy proteins that called PRPC to a PRPSC, and here you have illustrations of depositions of PRPSC assemblies on the brain that was revealed by theophlavine staining. So the prion pathology has triple determinism. It can be sporadic. In Europe, it represents one or two cases per year per million of people. It's represent 90% of the prion disease in Europe. It can be infectious, so you have the illustrations of Kourou that was kind of prion pathology that was transmitted by cannibalism. You have the variant of Creutzfeldt-Jakob disease. It's in Europe. It was the case for BSE mad cow disease. And it can be also iatrogenic. It means that you go to the hospital for a banal chirurgy, and as the instrument was contaminated by prions, 20 years later, you start to develop prion disease. And here in Europe, we had some case about 800 people that got prion disease due to because they received a contaminated growth hormone by prion. It can be genetics. So in a human PRP, you have more than 30 mutations that can induce the PRP disease. You have the familial CGD, Creutzfeldt-Jakob, that's due to mutations. You have another one that's Grashman-Strauchel-Schenker disease. And you have another one this one is quite interesting because it's called fatal familial insomnia. People don't die due to the prion, but they die mainly to the fact that they cannot sleep by tiredness, in fact. So you have this kind of this triple determinism. So the model of prion replications, the mechanism of prion replications, you have two models until now. And I will extend much more time on this aspect later in the presentations. You have the model of Griffiths. Griffiths was a mathematician in 1967 that just by observation of scrappy disease, he proposed just by using his observations that you have a autocatalytical process, A being the normal form of the protein, prion proteins, PRPC, and B being the PRPSC. PRPSC come and induce conformational change of A according to a autocatalytical process that's for who that did a little bit chemistry look like Brusov-Zhabotinsky reactions mechanism. There is another one that has been proposed in 1993 by Lansbury and Koge, and this one is more or less like tubulin or flagellin's growth. It's based on nucleation elongations. This model is more or less the one that's widespread in prion field, but we will come to this point later, is not really representative of what's happening in real life. So the prion dogma can be summarized through these very, very simple schemes. A first step that's correspond to a templating process. You have PRPSCs that induce the conversion of PRPC, and you have elongation or formation of assemblies that elongate. But as you can see, this templating step is totally inefficient. If you look, if you look the templating interface, you keep only one templating interface. And by analogy to what have been found for fungus prions or other type of prions, fragmentation process has been introduced to render to account the multiplication of templating interface and the phase of amplifications, and this corresponds to the amplification state. So the overall prion paradigm can be summarized by through this three-step: templating process, amplification by fragmentations, and spreading. Just one important thing: in mammalian prions, this fragmentation step is not yet established because all the events occur out of the cells, and no chaperones, no cofactors that can induce fragmentations has been yet discovered. But you will see that this problem can be overcome from the intrinsic and dynamic of prion assemblies. A few words about the PRPC, the normal prion structures. When you look the structure that has been resolved by NMR and X-ray crystallography, it's something very, very unusual. You have an terminal part that's completely it belongs to it's disordered. It has ability to bind copper or nickel's ions. The globular and it's not involved in the formation of the core of prion assemblies. The globular part, it's composed by two small beta sheet, two alpha helix that are in antiparallel, and a small alpha helix here. And PRPC is attached in the cell surface of a membrane through a GPR anchor that's attached in his C-terminal part. And something that I forgot to say also, that PRPC is a glycoproteins. So you have two glycosylation sites that is on this H2 helix. Concerning the PRPSC structures, so recently and since 2020, we have more or less 4 other cryo-EM structures. We had the cryo-EM structures of these PRPSC assemblies purified from brain of infected animals, and when also we look these structures, is we can see that all is in beta sheet structures. It's in what we call parallel in resistor beta sheet. And from one strain to another, now we have for at least 3 different strains, there is no significant change between the structures. We have only a few difference in the structure in this part. So it can render to account what we call the strain phenomenon. Also, a rapid view about what we call prion strains. I think that Ronald probably talked about this, and others that talk on prions talk about strains. But in mammalian prions, what we define as strains is that nothing changed in the primary structures of the proteins. Only things that change in the structure of the assemblies, and the difference of the structure of these assemblies is enough to induce difference in the biochemistry of PRPSC, the incubation period, and the physiopathology of the disease. Here you can see, for example, 2 different strains on the same primary structure of the PRP proteins, and you can see that you target different region of the brain of the infected animals. And here, as I told previously, you have the structure of 3 different strains, and the only thing that change is the angle between these regions and these constant regions. If you have questions during the talk, you can interrupt me, and it's going to be much more easy if you want to make things clear. So one thing that also in terms of protein folding that have to be underlined, that one given primary structures of PRPC can replicate multiple strains. Here is some data from the lab. It's a sheep PRPC proteins that can replicate more than 20 different prion strains in a stable manner. So it means that one given structures, PRP primary structure sequence, can give at least 20 different structurations with 20 different strain phenotype, time of incubation, lesional pattern, histopathology, and biochemical difference. So it indicates that PRP protein is a multi-stable protein in thermodynamically talking. So the state of the art in 2023, what's the main questions in prion fields? So the anatomic site of prion replications, we know that it's mainly in the central nervous system, and also it can occur elsewhere that PRPC protein is expressed. The subcellular site of prion replications, at least there is two schools. One said that the replications occur in the endosomes during endocytosis process, and others say that it's occur in the cell surface. But actually, the school that's gain in notoriety is more in the cell surface. Mechanism of neurotoxicity, until now, after 20 years of investigations, nobody knows how it occurs. Someone said it's the oligomers, like in Alzheimer's. Others say it's the prion assemblies, but nobody knows because there is any correlations between prion replications and cytotoxicity. The cytotoxicity occurs at the latest stage of the disease, so there isn't any real correlation between replication and toxicity. So the PRPSC structures, now it was resolved by cryo-EM, and we now have good idea of how the prion assemblies are really. and the mechanism of prion conversions. For the moment, the best that we have, this is the two that I show you, the one that Griffiths and the one of Landsberg. And the dynamic of the prion assemblies and how the structural diversification, prion mutations, such kind of things occur. Nobody know how it happens. And still are open questions. And how prion are amplified and spread. This also is something that's fully investigated. And it seems that in the field, I'm working on prion field since 20 years, and seems that people turn the eye from this subject. And if we have time, I will come to this subject at the end of the presentations. So in my lab, we focus on these two blocks of subject, the anatomic side of prion replications, the structures, and everything's relation between the dynamic of prion assemblies and the process of disseminations and replications. So to start, let's talk about the dynamic of prion assemblies and how structural diversifications can take place. So the prion replication, the actual dogma, and also is the same for in Alzheimer's and Parkinson and other amyloidosis, is okay, you have an assemblies and you have monomer that come and stick on the extremity, and you have elongations. And when you look on these minimal schemes and you try to do some biochemical informalizations, you see that it's something very linear and quadratic and something very banal. And when you go back to the structures, you can see that the assemblies are very stable, because now as we have the structure of the PRPSC's assemblies, we can estimate more or less accurately the stability of these assemblies, and we have something around 2000 kilojoule. That's per mole of the interaction between these different protomer. That's something very strong, and it's not let place to any intrinsical or constitutional dynamicity. So to first investigate such kind of things, a few years ago, in 2018, we started to set up a pipeline to go how it behave extractive prion assemblies extracted directly from the brain. So what we did, that we take a brain homogenate from the animals, infected animals. We solubilize the brain homogenate with some soft detergent, diethylmaltosil or sarcosyl, and we incubated at 37 degrees for 4 hours, and we loaded them directly in size exclusion chromatography, and what we obtain that we get a very small object, something that we characterized. I will come to this point later. By static light scattering and by calibration of columns and different techniques. And we obtained something that was about tetramer and a dimer of PRP. We noticed that as soon as we dilute these assemblies, just by simple dilution by factor 2 or 3, we displaced these assemblies toward the dimer. Or if we reduce the unique strength, for example, we are going by 200 millimolar sodium salts to 20 millimolar, we got large assemblies. So these simple experiments show that the PRPSC's assemblies can be splitted in very small oligomers that we call it SUPRP for subunit PRP. And of course, we try to see if we determine if they were infectious or not, and they were. All these assemblies were fully infectious, and with 100% attack rate, exactly in the same way that the crude brain homogenate could be. So we concluded that this small object constitutes the smallest infectious particle that we can obtain from prion assemblies. So it was quite weird, because we have to deal in one part we have PRPSC assemblies that has been described to be very deadpan PRPSC's assemblies, but just by sample dilutions and or by some simple solubilization in detergent, we got something that was soluble. So we established some minimal schemes that describe this assemblies process to something that's we call subunit PRP. We established also in the lab some in vitro replication process by using static light scattering as readout of fibers or assemblies growth. And here in these very simple experiments that I will describe, so we take PRPSCs, purified PRPSCs assemblies from the brain, we put it in a cuvette, we added monomeric PRPC, and what we observed, so you have an increase in the mean average molecular weight as we monitor it by static light scattering, and what we observed that so we have an increased phase and a decreased phase, and during this decreased phase we have occurrence of a very we have occurrence of oscillations. So as soon as you have, it was with 263K prion strains, and here it's for VCGD strains. You have exactly the same things. And as soon as you have such kind of oscillation that take place, it means that the replication is not a nonlinear process. It means that the simple addition of monomer in the extremity of the assemblies is not a good model to describe the prion replications. And oscillations also indicate that we have a dissipative process that involve a network of exchange between different PRPSCs subpopulation that exist or created during the replication process. So these simple experiments revealed that we have an incompatibility with a simple process of nucleation elongation process or end elongation process. So to investigate a little bit more in detail how the replications occur in vivo, I mean not anymore in vitro, but in vivo, what we did that we said, okay, we're gonna take some transgenic mice that replicate in bona fide prion assemblies. We're gonna inoculate them with the given strains, prion strains, and we're gonna sacrifice at different given time and and look the size distribution of prion assemblies from the T0 until the end stage of the disease. So I will focus only on this because it's going to be much more percutant. And as you can see that at the early stage of the prion replications, we have a small size distributions. So here is a small object, large object. We analyze the size of the assemblies through sedimentation velocities by centrifugation sedimentation velocities. And during the early step, we generate a small, very small object. And as the replications continue, we had large formation of larger assemblies. But what was amazing, it was the fact that the distributions was a bimodal distributions. It means you have first a small object that disappear in favor of large object. You don't have a translation of this peak from small to large object. So this was quite unexpected and unusual, and we decided to see if object that we have here, that we call it PRPSCA or PRPSCB, were the same or not. Previous to investigate this, we look in which conditions PRPSCA are generated and transformed to PRPSCB, and we noticed that when we removed PRPC, I mean we take PRPSCA, we put it in vitro, and we added PRPC, we have formation of PRPSCB. But if you don't put PRPCA and in the presence of PRPC, normal PRP, you don't have these transformations. So it means that the transformation of PRPC A to B require the presence of PRPC normal proteins. So it was something that was a secondary, what we said, the secondary templating pathway. And something else, that the transformation of A to B was something that was follow a sigmoidal shape, means that something that was enhanced by the formation of the presence of PRPSCB. So the formation of discrete, we had first the formation of discrete prion assemblies and a bimodal kinetic, as I said, and the transformation of A to B require PRPC proteins. And I missed something in this slide because I forget, I changed this this morning. We tested the activity of A and B, and we demonstrated that PRPC-A is much more replicative than B. So as the specific infectivity is the same, we concluded that the structure of PRPC-A and B was quite different. So then we decided to see, okay, we have two populations. Let's go, what's these populations? So we purified A and B. We were not able to purify that at one point A and B independently because the amount was low. So we set up a protocol to purify them together. And we analyzed this by atomic force microscopy using a 10 angstrom cantilever probe. And as you can see here, you have a teeny fibers at about 4 nanometers height, and stick on these fibers. You have a small spheric object that you can see here. So here is the topography picture, and we are able also to functionalize this cantilever with antibodies. It was in fact nanobodies against the N-terminal part of the PRP, and transform the adhesion curve into image. So the halo around the fibers that you can see is the N-terminal part of the PRP. So this small spheric object interact with the N-terminal part of the PRP. So it's the merge of these images. So we had two populations: the spheric object that was probably PRPSCA, and B, that's the fibers assemblies. So these two populations coexist and interact together. We characterized in a better way using Nano ER technique. So for those that are not familiar with Nano ER is an IFM technique that allowed you to look at single assemblies level, the secondary structures by according to the infrared response, the local structures of the assemblies. And we noticed that, okay, the assemblies had at least two different structures, something that's very, very structured beta sheet structures. And on these assemblies, you have some other objects that are on these assemblies that are much more less structured. And this is probably the spherical object. So this Nano ER approach led us to show that, okay, the assemblies are structurally heterogeneous. There is some intrinsically heterogeneities. So taking all this together, I mean, the fact that we have heterogeneity, we have two populations, and the fact that we have a bimodal evolution as a function of time during the prion replications. So we set up a minimal kinetical schemes that here you have this minimal kinetical schemes. We go and we do some modelizations to see simulation to see if we can render to account this complex evolution and this bimodal evolution during the replications. And Okay, even if the result was not so much accurate, it was not too bad, and we had a bimodal simulations, the bimodal kinetic evolution that render not perfectly what we observed experimentally. So to summarize this part, we demonstrated that the early step of prion replication generate at least two type of prion assemblies that we call it PRPSC-A and PRPSC-B. PRPSC-A is small oligomeric assemblies, and PRPSC-B is correspond to larger assemblies, the fibers assemblies, and these two objects interact together and co-evolve. And the conversions of A to B require a presence of PRPC that we define as the existence of a secondary templating process. So the coexistence, now the second part will concern the dynamic, the intrinsic dynamic of PRP assemblies. So as we had two different type of PRPSC assemblies, the questions was, okay, in the context that we don't have replications, how these two structurally distinct assemblies could co-evolve within the same environment, and why they are maintained together. Because we are expecting that, okay, if PRPSC-A is transformed to PRPSC-B, at one point they should disappear. And what's the relation between A and B in the absence of any replication context? So for this, we did a very, very simple experiment also. The experiment consists to purified infectious prion assemblies, put them in the cuvette, and look how they evolved by light scattering, so the mean average molecular weight, as a function of time. And what we observed is that during this, the assemblies, the prion assemblies spontaneously disassemble. So we have a first stage that is very slow, and a sudden depolymerization, like a catastrophic depolymerization. And during these catastrophic depolymerizations, we have occurrence of oscillation that occur. And when we do a FT, Fourier transform of these oscillations, we have a period around 3 minutes between the period of this 3 minutes for these oscillations. We investigated a little bit more these oscillations as a function of initial protein concentrations. So at large, when we increase the assemblies concentrations, we have much more oscillations. At lower concentration, we have less oscillations. Here you have the SLS, the static light scattering evolution as a function of time, and this panel represents you the envelope of this curve to make in exact the oscillation. And here you have the ATP oscillation of the zoom of these regions. So what indicate these oscillations? It means that the assemblies are far from the equilibrium. If you have oscillations, it means that the objects are not in a stationary state, they are in oscillation. And I think that Marie, in her presentations, come to this point a few weeks ago, and I will detail a little bit more this part later. It means also, if you have oscillation, you have exchange between the different subpopulation. If you don't have exchange, you cannot have oscillations. Is a question of physics. And if you have oscillation, it means that you have multiple catalysis that occur. So the objective was, okay, what's the mechanism of this exchange process? We did also some perturbation relaxation approach to see these oscillations and to characterize this oscillation. Is a temperature ramping perturbation. I will not extend on this point because I will not have time. But also here it demonstrates that we have exchange process. So to characterize this exchange process, we take advantage of Nano ER techniques and the fact that we were able to generate bona fide infectious assemblies labeled by C13 or C12, because in infrared we can distinguish spectra from C13 labeled assemblies from C12. So we generate the same strains in C12 and C13. We mix them. We incubated at 37 degrees, and we did Nano ER spectra. And we observed that at a single assemblies level, so in green you have C13 assemblies, and in pink you have the C12. And if you have pink on green, it means that you had exchange process. And here, as you can see, you in the reconstitutions images, you have pink on you have green on pink, and it means that we evidence the exchange process at the single assemblies level. So we took all this data together. We had a lot of exchange with the mathematicians group, Marie, and taking to the fact that we have a spontaneous depolymerizations of PRP assemblies, and in one hand, we have two subpopulations that coexist in the same environment, and they are generated during the replication process. The fact that A assemblies interact with B, as we show it by IFM, and what Nano ER also indicate, we established a minimal kinetical scheme that render to account this the dynamic of these assemblies that can be represented in this way. You have a catalytical depolymerizations, means that you have a object that interact with assemblies and induce their depolymerizations, and in the other part you have an interaction between A assemblies and some subunit of fibers assemblies that make A transformed to B. So when you have this, we go and we try to do some simulations to see if we are able to render to account this complex system. So for such kind of complex system, it's an illusion to be able to describe it fully. So we get something that was not so much bad. So here you have simulations. Here you have an experimental data, and here you have the simulation with a first stage that was very slow, and the oscillation that occur, and these oscillations correspond to the transformations of A to B and B to A, and increase of the polymerization, the size distribution of the assemblies. So to finish this part, and if I have 3 minutes more, Lucy, I can have 3 minutes more?
Yes, you can.
Yeah, okay, thank you. I will make some recapitulative here, and I will probably talk about what's the consequence of this in terms of these schemes in terms of disseminations. So we demonstrated that the replication process is intrinsically generate diversity. PRPSC assemblies are highly dynamic, so the idea of that you have a deadpan assemblies is completely is not adapted to mammalian prion assemblies. It exists a complex process of exchange between PRPSC's subpopulations. The scheme that I show you is probably the very simplistic, but at least it describes the process of exchange that could occur at a molecular level, and so the existence of this dynamicity can allow us to try to develop some perspective to investigate the dissemination process. I told you at the beginning that there is any fragmentation process that has been described in mammalian prion assemblies. But here you can see that as prion assemblies are intrinsically dynamic, you don't need any more to have fragmentations. their sample dynamicity is enough to try to describe the to propose a mechanism for the disseminations. And this is what we are doing actually. I will not extend to this, but we adaptate these minimal schemes in terms of we added a spatial component. We take advantage of the existence of a lot of biological data concerning the response of neurons to prion replication in terms of unfolding protein response, how PRPC expressions are affected during the replications. We put all this together, and we are exploring how we can simulate the spreading process during the prion replications in an organoid made environment, or in what we call organotypic slice, and how pattern could appear according to this autocatalytical process, and a pattern like Turing pattern. So thank you very much for your attentions, and I would like to thank all the participants to all this project, my groups, the group of Marie that we exchange a lot for all the simulation mathematical modeling, the University of Concepcion for all the organoids and all the things that we cannot do anymore in France due to prions restrictions, and Nano ER, such kind of things that we develop a lot with Ariane Denizet, Alexandre Desi, and Jeremy Mathieu. Thank you very much.
Thank you very much, Human. So we have already 3 questions, but if you have more questions, please type it in the Q&A. So the first questions are from Christian Grissinger. So I'm going to read it for you, but you also can find it on your screen.
Activate my chat, yeah.
So you show the different assemblies. It seems that the structure of each protofibrils is the same, but just the quaternary arrangement is different. This is quite amazing. So you don't see any difference faults, even from different sequences.
This is a very good question. In fact, okay, I have to be honest with you. The cryo, I mean, if you are talking about the cryo-EM structures, am I right?
Well, I think, yeah, okay. I think so.
I will assume that it's what you are talking. Otherwise, I will come back to your questions later. Yes. Okay. Yes. The cryo-EM. Okay. I will again show my presentations. The authors that did the cryo-EM structures for different prion strains. Said, okay, there is a difference, but the difference resides in these regions. But honestly, and I think that the difference should reside much more on the dynamic of the assemblies than just on these simple variations. They claim that this difference is enough to determine what's the strain phenomenon. But okay, as we have only for 3 different strains and we don't have much more, we cannot be able to say, okay, it's coming from this region and not others. Okay, but I cannot how to say argument it much more.
So it looks that the prion is very, very different from, for example, alpha-synuclein and tau and so on, which have protofibril, I mean, which have big difference in the protofibrils depending on from which brain, from which diseased brain they are isolated and so on. Is that the case?
Yes, I know that there is a lot of authors that claim, okay, you have if you take from prion alpha-synucleins or tau. Let's talk about tau because I'm much more comfortable with tau than alpha-synucleins. People or also A beta, they said that, okay, if you take, for example, A beta from frontal cortex or rather than other regions, you can have a different conformation of these assemblies. This is true. This is true, but something that people forget and should, have in mind that in brain, it's not the same for prion proteins. A beta, you don't have only A beta 42 or A beta 40. You have a mixtures, and nobody go and look what's the ratio of this A beta 40 or 42 in the assemblies that they are characterizing, and they said that they have. It's the same for Tau. Tau has more than 96 site of phosphorylations, and this is not something that's there is a continuous in the Tau assemblies, misfolded assemblies. So they could be due to the different phosphorylations, such kind of thing, this structural difference. In prions, and it's, we turn mad when you are working mammalian prions, you are turning mad. You have one primary structures, only one primary structures, and you have only, you can propagate more than 20 different structures in a stable manner. It means that you can do more than, let's say, 7 different passages in transgenic mice. This is what we do usually. And they don't move. They don't have mutations. They don't switch. The structure doesn't switch. You have the only way that they switch if you change the host. If you change the primary structure of PRPSC.
Okay.
So coming back to, sorry, go.
I don't want to dominate the discussion totally. I had another question regarding the oscillations, which are really, amazing.
Yeah, this is.
And did you find ways of modulating the frequency or so, maybe by temperature, by
yes.
whatever?
Okay. I didn't want to extend time with this. This is a temperature ramping perturbations. What does it mean? So we did a cyclic up, down, up, down, up, down. And during the phase of up, you see that the frequency change. So it seems that you have, if you come back to the fast diagram, it means that you increase the reactions process. So what means that you, by Arrhenius, you change the rate of the constant. By adding salts, by increasing the ionic strength, you reduce totally the frequency of these oscillations. By decreasing the ionic strength, you increase. We arrive to have at 20 millimolar sodium salts, we arrive to have a period of oscillation of 4 hours. 4 hours. We are quite glad because at the beginning when we had this, and I did a talk to physicians. When I saw it, I showed the relaxation, these experiments. I said, okay, come on, it's the 50 Hertz that's coming from the surrounding environment. I said, okay, no, it's 2 minutes period. And now that we have 4 hours period, it's definitively something that occurs in real life. In real life.
Thank you. I don't think there is another question. You had a third question, Christian. You don't want to ask it? No, it's okay. It was about, you asked if there is any isolated from human brains of people that have died of CGD. Yes.
In fact, the experiments that I show you here coming from these experiments, it's coming from VCGD's PRPS assemblies purified from human brain.
Okay. So thanks again, Human. There is no more question. If you have more questions during the second talk about human talk, you can still type it in the Q&A and we will answer it after the second talk.
Thank you very much.
Now we are going to, you're welcome. We are going to switch to the talk of Sven. So Sven is a research director at the CNRS in France, in the University of Bordeaux, in a lab in the University of Bordeaux. Sven got his PhD in Bordeaux in biology in 1993. He did the first postdoc in Vancouver in Canada and the second one in Montpellier in France. And Sven studied fungi, and more particularly, he focused his work on the domain ETS from the domain ET, from the gene ET, and which was ETS was involved in amyloid forming process in fungi and in bacteria. So Sven, please.
So I share my screen. I just check that that you see the full screen.
Yeah, we see the full screen.
Okay. So thank you very much for the invitation, and I'll just like to say also that I was amazed by Humans talk, which I really found great. So I'd like to talk about amyloid signaling motifs that occur in fungi and in bacteria, and that regulate cell death in these microbes. So as we see from the biological point of view, we can classify amyloids as either being pathogenic that lead to a variety of neuronal or systemic diseases. And then we also have functional amyloids that have a range of functional roles, both outside and inside of cells, both in prokaryotes and in eukaryotes. And those roles can range from structural scaffolding. They can be connected to hormone storage and release. Amyloids can be involved in cell walls and envelopes and biofilms. They can be involved in cellular memory, and they can also act as microbials, as the work of Michael Landau, for instance, illustrates. And I would like to talk about roles of functional amyloids in the context of signal transduction. And so that would be the outline of my talk. I will need to go back to the HEDS prion system, because that's how this whole project kind of started. And then I'd like to talk about amyloid signal in the context of node-like receptors, and describe different amyloid signaling motifs in fungi, and also show that this process can be extended also to bacteria, and make some comparison between amyloid signaling motifs in microbes and those that occur in innate immunity in animals. So the HEDS prion was discovered in the context of a phenomenon, the study of a phenomenon that is termed vegetative incompatibility. I have to bring you through this because it allows to explain how these things work. So in fungi, there is a very common process that occurs when two strains meet. They have, filamentous fungi have the specific property of fusing their somatic cell. That's not something that you see very often in living beings, but if you confront two strains on a medium, on a plate, very simply, they will fuse their somatic cells and make a heterokaryon, so a cell that contains a mixture of the two strains. And there can be two outcomes to these encounters. Either the strains are compatible and you will grow a mixed organism with the two nucleotypes, and then there's another outcome if the strains are incompatible, where right after the fusion event, the fusion cell will undergo regulated cell death, and you will see this demarcation line between the two strains that is termed a barrage, and that's how it actually looks in real life. If you it's something that is very common, that has been found in basically every species where it has been looked for. And maybe you know of these objects that are called spalted wood, that are sometimes, it's something that is used to make nice artifacts like bowls or musical instruments. And when you see those dark lines in the wood, these actually correspond to incompatibility reactions between fungal strains that colonize the wood. So what is it good for? What's the function of this incompatibility mechanism? Fungi, like all cellular organisms, have viruses. They are very frequently infected by viruses, but what is specific to fungi is that these viruses do not have a free form. They're not excreted outside in the medium. They're packaged. They have they are virions, but they're not excreted, and they are mostly, if not essentially, transmitted through these anastomosis events. So basically, if a strain is infected by a virus, if it fuses with another strain, it will transmit its viruses. But if the strains are incompatible, so if this fusion leads to cell death, then the transmission of the viruses will be restricted. This is illustrated here by this picture. It's taken from a species that is called Cryphonectria parasitica, which is a pathogen of chestnut trees, which basically ruined all the chestnut trees in North America. And you see that when the strain is infected by a virus, it gets that white phenotype, and it actually loses virulence. That's why the dynamics of the virus are very important. And if you confront a virus-infected strain with an incompatible strain, the virus will stay restricted to one strain. It will not be transmitted. If the strains are compatible, then the other strains will get infected by the virus. So very simply said, the incompatibility mechanism can be viewed as antiviral prophylaxis. So very much like the face mask that we used in the ongoing COVID crisis. So the genetics of incompatibility have been worked out in a number of species, and I will focus on this species here called Podosporan serina, which is our model species. And in that species, there are 9 incompatibility loci, so the genes that are controlling this process. And we will focus on this one here, which is called HEDS. And incompatibility occurs between strains that express the smallest allele and strains that express the largest allele. So it's 2 alleles of the same gene, small s and large s, and small s and large s are incompatible. That means that if you confront the 2 strains on a solid medium, you get the barrage line. Okay, so what is interesting about this system is that the small s protein is a prime protein. It exists under a soluble form, monomeric soluble form, and an aggregated amyloid form. And it's the amyloid form of the protein that leads to cell death when it's interacting by the product of the other allele. So if you fuse small s in a prime state with the largest strain, you get the cell death reaction. If the protein is in the soluble state and you fuse with large s, you get no incompatibility reaction. So it's this interaction of the prime form of one of the protein with the other allele that leads to the incompatibility reaction. And I just insist on that here. We have a process of prime formation and amyloid formation, but it's not connected with toxicity. So it's not the transition from the soluble to the aggregated state that leads to toxicity. Those aggregates are the cells can express high amounts of those aggregates, and there is no problem of toxicity. And so this process, this B-stability of soluble to the aggregated state, makes this transition epigenetic switch. So this prime becomes a genetic element, and all the progeny of cells that express aggregates will express aggregates. So you have basically this B-stable state that can be inherited both mitotically and meiotically. So this protein forms amyloids, and it's actually a 2-domain protein. So that has been worked out in particular a lot by the group of Roland Riek at the ETH Zurich. So we have 2 domains. Oh, and I should say that the amyloid fibrils display prion infectivity. That has been shown also in the case of the PRP prions. So if you reintroduce the fibrils into the fungus, you will get the switch from the soluble to the aggregated state in vivo. So it's a 2-domain protein with an N-terminal folded domain and a C-terminal natively unfolded domain. So as we talked about PRP right before, it's actually the opposite situation as with PRP. It's the natively unfolded part of the protein that is the one that then goes transition to the amyloid state. And the amyloid, the structure of the amyloid state was published in 2008 by the group of Beat Meyer at ETH Zurich. And it turns out that the protein, so the structure is a beta solenoid with 2 layers of beta strand per subunit. And the prime forming region is actually composed of 2 pseudo repeats that we term R1 and R2 that each compose 1 layer of the beta solenoid structure and they are connected by a flexible loop of 15 amino acids. And what we have on the structure here is it's stabilized by 2 asparagine ladders. It has mainly hydrophobic residues in the inside of the structure and charge or polar residues at the outside, which sort of explains why these fibrils are not a problem for cellular viability, because in a sense, what it's exposed to the cell is very normal with an hydrophobic polar interface towards chaperones or other cellular control mechanisms. So what about now the incompatibility reaction? We talked about this transition from the soluble to the aggregated state, but how can we explain incompatibility? And what actually happens in the incompatibility reaction is that the C-terminal part of the small s protein templates amyloid conversion of the protein coded by the other allele, large S. And this amyloid transition in the C-terminal region of the protein induces a conformational change in the globular domain that we term the hello domain, and that expels N-terminal hydrophobic alpha helix that goes to the membrane and leads to membrane damage and a subsequent cell death. And that's, again, has been worked out a lot by the group of Roel and Riek at ETH Zurich. So that's what you see, for example, in this image here, where you have a strain that expresses a small s fused with GFP that undergoes fusion with a strain that expresses large s fused with GFP. And you can see that in the contact zone where the strains fuse, you have relocalization of the largest protein to the cell membrane. What is interesting is that the hello domain, so the domain that induces cell death, shows a limited but significant homology with plant CC domains and the effector domain of a protein that is called MLKL. And both of these proteins are involved in regulation of immune cell death, both in plants and in animals. And what these proteins have in common is a folded alpha helical domain with an N-terminal hydrophobic helix, which very often has a negatively charged residue at position 2 or 3. So the hello domain, the plant CC domain, and the MLKL domain share the same execution domain for induction of regulated cell death. So once the prime-forming domain of HED-S has been identified, and the fact that it's composed of these 2 repeats, once that has been identified, we tried to devise a sort of a consensus sequence for this elementary motif, for the elementary repeat. So we aligned all the homologs of HED-S we could identify in the genome databases that were available at that time. And you can see that that's something important for the case of functional amyloids as compared to the pathological ones, is that you can make sense of amino acid alignments and conservation, because the amyloid state is the sequence onto which the evolutionary pressure is acting. So you can make sense of the evolutionary conservation. And it's what you see if you map onto the HED-S elementary structure, the conservation. You can see you have a very strong conservation of the asparagine that form the asparagine ladders, of that glycine here that forms the sharp king between the 3rd and 4th beta strand, and that you have a lot of hydrophobic residues inside that are also conserved, and that the conservation of the polar residues at the outside is relatively low, but so you sort of the conservation makes sense with the overall structure of the fibril. But where the surprise came is then when we use this consensus sequence to identify additional head-S homologs. And when we did that came the surprise, maybe one of the biggest surprise in my research work. The hit we found, the second best hit, was to a protein we call NWD2, and that is located right beside the largest gene in the genome. So it's 600 base pairs away. And that protein is actually NLR, node-like receptors, and I will explain what those are. That is part of a large gene family, which includes other head genes. So we were very surprised because it was very surprising that we should find a hit to the gene that is right beside it. And the region of homology occurs in the N-terminus of this protein, and it's present only once as opposed to twice at the end of the largest protein. So what are NLRs? NLRs are node-like receptors, which are intracellular immune receptors that work in innate immunity in animals and in immunity in plants. They are central players in immunity, both in a number of animal lineages and in green plants. There are also homologs of NLRs in filamentous fungi and also in bacteria. They are typically three-parted proteins with the N-terminal effector domain, a central nucleotide binding and oligomerization domain, and C-terminal repeats, which can be of the leucine-rich repeat types or of other types. And the way NLR receptors work, I will illustrate that with the ZAR1 immune receptors that is found in plants. You have these three domains, the effector domain, which here is the CC domain, the nucleotide binding and oligomerization domain, which is the central domain, and the leucine-rich repeat domain at the C-terminus. So their working process is a so-called ligand-induced oligomerization. So they recognize a ligand, which is, that's the way they work as immune receptors. They recognize a bacterial effector, and when they bind to this effector, or actually it's a modification of a self-protein by this effector, but essentially it amounts to the same. They will change conformation and oligomerize and cluster together the CC domain at the center of the structure, which is called the resistosome in plants and inflammasome in animals. And this clustering leads to expulsion of an alpha helical, an alpha helix, an hydrophobic alpha helix, which then targets the membrane and essentially turns this structure into a calcium channel and leads to membrane perturbation and cell death. So based on this model, what can we make of the homology that we have at the N-terminus of NWD2 and the C-terminal part of large S? We came up with the hypothesis that maybe this protein recognizes a ligand and then, like other node-like receptors, oligomerizes, and that this oligomerization would then be used as a trigger to induce the nucleation, the amyloid nucleation of that N-terminal region, which then could act as a template to activate the pore forming activity of the largest protein. So there would be actually two ways of inducing toxicity of the largest protein, either through the smallest prion or through the NWD2 NLR receptor. So I have to skip that, but we obtained in vivo evidence that this was the case, that if you use a specific ligand of NWD2, you can induce prion formation. So you can induce this nucleation of the amyloid structure. And we also verified that this region does indeed form amyloids and that those amyloids have prion infectivity. Quite interestingly, if you make proline mutations in this region, you have a peptide that can still form amyloids, but those amyloids lose the prion infectivity. So probably they're not able to form the beta solenoid structure that is specific for templating the smallest amyloid formation. So what we have there specifically is a mechanism by which a receptor induces activation of his effector protein, and this activation occurs through amyloid templating. So there is a proportion of the NLR receptors that we find in fungi that activate their effector domain by transmitting an amyloid fold. And what I think is important here is that this process is intrinsically a cross-seeding process, because you seed an amyloid motif from one protein to another motif, which is heterologous. There are, depending on the systems, quite a bit of amino acid differences between the motif on the receptor and on the effector side. Just want to check time a little bit. So we've looked for other motifs like that. So in addition to HEDES, we looked for other motifs like that in fungi, and we found a few. And I'll just mention some specific examples and what they, how they can make an interesting addition to this story. So the first one I would like to mention, we also found in Podospora. It's a protein we term ELF, and it occurs in exactly the same genomic organization as HEDES and NWD2. So you have a halo domain protein. So it's actually a variant. It's halo-like, and it's adjacent again to NLR, but with a different domain structure. We have TPR repeats instead of WD repeats, but it's also a NLR, and the two genes are adjacent. And you have two repeats of the motif on the HEL protein, on the HEL side, and one repeat on the NLR side. If you make a consensus sequence of the motif that you find in HEL, you see that it's quite different from the consensus sequence for the HEDES family. So you don't have the asparagine ladders, and you have basically conservation of the strongly conserved glycine that makes the sharp kink between the third and the fourth beta strand. And other than that, the consensus sequence are quite different, and the identity between the two amino sequence is in the range of 15% to 15%. So Antoine Loquet here in Pessac solved the NMR structure of this, if you want, of this variant of ELF, of a HEDES, which is called HELF. And what is quite interesting is that in spite of the very large amino acid difference, the structures of the two proteins is essentially the same in terms of the backbone structure. And in spite of this structural homology, there is no in vivo cross-seeding between HEDES and HELF. So that means that you can propagate either HELF prions, and you will not induce transconformation of HEDES, or the opposite, where you can have, so here HELF is labeled in green and HEDES is labeled in red. So you can have HEDES prions but no transconformation of HELF, or you can have transconformation of both, but they will form separate aggregates. So we have, in spite of strong conservation, no cross-seeding between those two amyloids. Another motif I would like to mention is a motif we term sigma, which is a little bit longer than HEDES. It's more in the range of 30 to 35 amino acids. And this was identified in the context of a study of another infectious element from fungi, which is called the sigma element from Nectria hematococci. It was originally described as an infectious element of unknown origin by Marie-Josée Daboussi in the 1970s, and later worked out also by Philippe Sillard. And it turns out that the sigma element, which appears as these abnormal growing sectors here that are pigmented with the purple structure here, if you do a subculture of this fungus, from time to time, you will see that growth alteration and then you can show that this growth alteration is infectious. So if you take a sample here of a strain in the zone where the phenotype is affected and put it into contact with a normal growing strain, you will transmit that phenotype. And it turns out that the sigma phenotype is due to this gene cluster, where again we have an NLR and two proteins that are termed CES-A and CES-B, where CES-B, there is a mistake here on the slide, CES-B encodes a lipase and CES-A encodes a distant homologue to a hello domain protein. So here we have a situation where a same NLR actually signals two different proteins, including an enzyme. And again, the three proteins are grouped as a single gene cluster that would, in a sense, look like a bacterial operon. And the last system I would like to mention is the PP motif that we also identified in Podospora serrana and in another fungus that is called Chaetonium. And this motif is characterized by this J, hydrophobic Q, hydrophobic J motif. We call it pseudo-palindromic because it's like a pseudo-palindrome. And what is interesting is that this motif is also what composes the core of the amyloid motif that you find in the RIP1 and RIP3 kinases, which are the regulators of necroptotic cell death in mammalian cell. And RIP3 and RIP1 form a kinase complex that is assembled through this amyloid motif. So what is interesting here is that you have two levels of homology in these cell death-inducing cascades in mammals and in fungi. You have one level of homology at the level of amyloid signaling, where you have short amyloid motifs that are used in a signaling process. But in the case of the mammalian system, this kinase complex then phosphorylates the MLKL protein, which then undergoes transformational conversion and goes and forms a membrane pore. So you have a similar situation in the fungal system, but the amyloid motif is different. It occurs at the end of an NLR and is then used as a template to activate the pore-forming protein. So I'd like to take notice of the two levels of homology that you have in these regulated cell death processes in fungi and in mammalian cell, where you have the same building blocks, but they sort of assemble in a pathway at different with different configurations. We've been interested by this homology by the RIM motif. So this amyloid motif in the necroptosis pathway is called RIM. And the fact that it appeared also conserved in fungi, we found that quite interesting, and we and others have noticed that this RIM motif is also found in some bacterial proteins. So we looked into those and found that interestingly, those occur. So this, for instance, in this operon here, and that they occur at the N-terminus of a gene that encodes a protein we call BEL, because it shows a remote homology with the hello domain, and in an adjacent gene, in this case, it's even an overlapping gene that encodes NLR with the NBR and TPR repeats. So you have the same configuration as in the case of the fungal NLR receptors and executioner proteins. So we made the hypothesis that maybe these amyloids, these motifs or RIM-related motifs in bacterial genomes might also be working as signaling devices. Here is a sort of consensus sequence of the BEL domain, and you found again a relatively hydrophobic region in the very N-terminus of the protein and the common conservation of a negatively charged residue in position 2 or 3. So what we did is that we fished out all the BEL domain protein that we can find, so several thousand of them, and then systematically look for a situation where those BEL proteins were residing adjacent to NLRs, and look for a situation where there was homology between the C-terminal part of the BEL domain and the N-terminal part of the NLR. And by doing so, we identified 10 what we call bacterial amyloid signal sequence or BAS motifs, and we grouped those into 10 families. And what is interesting is that we found those motifs exclusively in bacteria that form multicellular structures. So in actinobacteria that used to be called actinomycetes because they look so much similar to fungi, or in cyanobacteria that used to be called blue-green algae, although they're not algae, but they are bacteria. And also in chloroflexi, which are also non-photosynthetic filamentous bacteria. So there was a bias in the genomic representation of those amyloid signaling motifs in multicellular bacteria. Very much like you find the amyloid signaling motifs in filamentous fungi, but you don't find them in unicellular fungi like many yeast species. So there is something that about those signaling motifs that appears to be preferentially associated with multicellularity. So those are the, I'm not dwelling too much on that, but those are the different BAS motifs. They're all characterized by different consensus sequence where you find frequent recurrence of glutamine and asparagine residues. Just to mention the fact that those motifs are very well recognized by the ArchCandy algorithm developed by André Cajava in Montpellier. In our hands, for the detection of functional amyloid motifs, this tool works very well in contrast to many other amyloid detection softwares, which very often show a completely flat signal for many of the motifs that we then experimentally evidence as amyloid forming motifs. So we did some experimental work with those motifs and could show that. So what we did is ask, okay, we propose that there are the analogs of the fungal prion forming motifs and amyloid forming motifs. So because we knew how to do that, we didn't know how to work with filamentous bacteria, which is sort of an art of it, an art on its own. So we use those motifs and just ask simply, could they behave as prion forming motifs in a fungus? So which basically transport them in Prodospora and test if they would behave analogously to fungal prion forming motifs, and that would be the case. So in addition to that, they form amyloids in vitro, but you can make this kind of experiment where you express one of the motifs in one strain that is used as a donor, and then you confront it with a receiver strain, and you can just verify whether or not you will get a conversion of a fusion protein to an aggregated state in the receiver strain. So I'm kind of skipping that. What I would like just to mention, I mentioned before that some of those motifs are homologous to the rim motif as found in mammals. So we also treated the mammalian motif in the same way. We used the mammalian motif and tried to see if it could work as a prion motif in Prodospora, and that turned out to be the case. So then we had a system where we could look at cross-seeding between the bacterial, fungal, and mammalian motifs in vivo, and to see if they could cross-induce each other. And so we found that this was actually the case. So if, for instance, you use a PP motif as a donor, you can use a transconformation of a rim motif. So the bacterial, mammalian, and fungal motif all cross-seed. But I should add that the cross-seeding is partial. So if you cross-seed, for instance, the two mammalian motifs, the RIP1 and RIP3, between them, you get much higher rates of cross-seeding than, for instance, between a bacterial and mammalian motif. And this is just the structure of the rim motif as published by the McDermott lab, where you can see that this JVQVJ motif is the core of the amyloid motif. So apparently, and that's basically the only region of homology that you have between the mammalian, bacterial, and fungal motif, and it also occurs in a Drosophila rim-like motif, and maybe this motif is sufficient at least to have some level of cross-seeding, transkingdom cross-seeding between those motifs. So in the end, we have amyloid signaling that occurs both in bacteria and in fungi, where you have a number of different motifs, at least 4 in fungi, now at least 11 families in bacteria, and those can signal a number of domains, including the halo domain, but also other domains with enzymatic activity. And I would just like to dwell a little bit on the amyloid signal process and how it can be compared to other, signal transduction process. For instance, we have to compare it with a situation where the receptor and the effector would be on the same protein, because basically what happens in many NLR receptors is that the halo domain is directly attached to the NLR part. So that's the architecture we call all-in-one. So you can dispense of the signaling way, because you have the cell death effector protein that is directly connected to the NLR. But this imposes that the stoichiometry between the effector and the receptor is set to 1. Whereas in the case of the amyloid signaling, you have 2 advantages. You can disconnect the stoichiometry of the receptor from the effector. And then obviously one thing that is interesting is that you have signal amplification, because the effector, once it is transconformed to the amyloid state, can act as an inducer for more transconformation. Then we also have the possibility with one receptor to turn on different effector proteins. You have seen those examples where you have one NLR and then several effector proteins that are encoded by the adjacent genes. So it can go from one receptor to several effectors, which can then also cross-activate. You can have the opposite situation. There are also situations where you have several receptors that converge on the same effector. And another interesting situation is the case where you have partial cross-seeding. So you can have, with different amyloid motifs, you can have either branching or tight signaling cascades that go from one receptor to one effector, but where you can also have cross-activation from different effectors. And what we have to do also is to compare this mode of signal transduction with what you could have by let's say, phosphorylation, or just, let's say, a proteolytic cleavage to activate a protein. So that will give you the same kind of advantages as the ones I've described here, but you have the advantage of having very short motifs in the range of 25 amino acids, and no, let's say, energy expenditure, because you have no covalent modification. The energy you take from the system is just the energy that you bring out of the actual amyloid folding. So those were the conclusions, but I think I've been through that. One thing I'd like to reiterate is that this process is inherently a cross-seeding process, and the identity between the signaling motif and the effector side is often in the range of 60 to 30%. So you don't need to have identical sequences for this system to work in vivo. And I would like to thank people involved here in my group, or previous members: Hassan Daskalov, Virginie Coustou, Corinne Clavé, Alexandra Granger-Farbost, Léa Barlosa, and our collaborator Antoine Loquet in Pessac, Witold Dirka, Wrocław University in Poland, and previous collaborators Ronald Rick and Beat Maya.
Thank you very much, Sven. If you have any question, please type it in the Q&A and I will read it for you. I don't know if someone, no, there is no question right now.
Human, can I ask the question? Or should
yes, of course, you can ask the question, Human.
Oh, it was a very, very nice talk, Sven. In fact, if for the cross-seeding, I mean, it's not a question about the cross-seeding, but you think that you can have two different type of assemblies with the same effector. I mean, two different assemblies have different effector interaction with different effector interactions. Am I clear or not?
I
For
think
example, yeah, tell me.
so let's take the example of Podospora. In Podospora, there are three systems like that, where you have an NLR and an effector. And the three of them, so they essentially they have the same kind of effector, but it's three different operons, if you want. I'm using the bacterial word just to make it simple. Three different clusters. Each one has its receptor and its effector, and there is no cross-seeding between the motifs, but there are also different motifs in terms of sequence. One is PP, so it doesn't have anything to do with HED-S, and the other one is related to HED-S, but as I showed you, they're very different in terms of sequence, although they're very similar in terms of structure. So there in Podospora, it would be three tight, independent pathways. But we know of other systems because of the genomic structures where you would have conversion of different receptors located in different loci in the genome, but they would converge on a single effector.
It's very interesting because it's referred to what you can call amyloidome, in fact, within a cell.
That's right. That's right.
Thank you.
Sorry, I have just a very quick question for human. Okay, you appeared again, disappeared. So I enjoyed your talk, really great talk. My question is, some people are proposing that besides prions, other amyloid-forming proteins, okay, under specific environmental condition may be infectious. This may have very relevant consequences because we could talk about transmissible Alzheimer's disease or transmissible Parkinson's disease. I would like to know what is your opinion on that.
You know, it's a very, very big debate. I mean, start to emerge in almost when Sven and me started to work on prions. I mean, what makes prion, and what's the difference with other amyloidosis? I mean, Sven can answer this much more better than me, but for me, I mean, what makes amyloid be prions? Be able to replicate adaptations, because adaptation is very important, because from different environments you can have different form of object, and especially what are the process of amplifications. For example, when you go for I mean, is the case for HED-S or other cytoplasmic amyloid, you have cofactors that contribute to induce the fragmentation, you know, the phase of amplifications. If you don't have this, you should find an alternative process to this. So it means that the assemblies should be either not so much stable in order to spontaneously fragmentate, or have what we can envisage some autocatalytical fragmentations. I didn't extend on this subject because for a question of time. I show that you have A that interact with B. This interaction is not just for interactions. Could induce what we call entropy by entropic pooling, some spontaneous fragmentation. So it means that during prion replications, PRP generate two kind of assemblies. One are fibers, and one it's fragmentas, its own fragmentas. You see? So what make prion in assemblies prions, to summarize, to address your questions, is something that have all the informations to auto-perpetuate.
Okay.
And only an amyloid, a very stable amyloid, is not enough.
Thank you.
I don't know if, what do you think, Sven?
I might just add one thing a little bit on the side, that that one thing sticks out in the case of PRP is the level of infectivity of the fraction you have. That's something that you know, if we extend the analogy between PRP and the other amyloids and all those cryo-EM structures that come, and if we tend to blur it all into one thing we have to remember that is absolutely fascinating and frightening in the case of PRP is the level of infectivity you have, and that is a direct explanation for why you have the naturally occurring diseases in so many animals, whereas the transmission of the other amyloids, tau, A beta, Parkinson, you can show infectivity, but it's in an experimental setting. So there still is something absolutely crazy about PRP, and that's the level of infectivity, and that's why, you know, there remains something really specific in the case of PRP that requires, I mean, it stands aside in spite of all the structural analogies you can make with the other amyloids.
Yeah, this is true. Yeah, this is, and also the mechanism. I think, you know, people say, okay, as soon as you have a seeding phenomenon, seeding, and you know, the Lansbury mechanism of a simple Becker-Doring's mechanism. As soon as they have this, they said, okay, should be a prion-like. They use the terminology prion-like, but the terminology prion-like in terms of physical chemistry, it means that okay, whatever you do, the system will evolve to the amyloid. The only things that you do is just to enhance the process, to accelerate the process. So it means that the system is kind of in supersaturate. It's only to put a dust to initiate it. It's why also I think that this kind of scheme is very, I mean, it's joined a little bit what you present, kind of cross-seeding. So it means that your assemblies, your system is already in a metastable situations. The only thing that it needs, something that initiate a amyloid pathway and go to induce the formation of assemblies.
This is a quite fascinating subject. If I understood correctly, it's something like saying that some proteins that are not totally intrinsically unstructured, disordered, may hold some information. That's why alpha-synuclein is considered a bit more risky, dangerous than amyloid peptides. Is it true?
Yeah, could be. I will just put some nuance in this. PRP, it's folded.
Yeah.
Okay. So it means that it could exist at least. Don't forget the plethora of strains that you can have. You have PRPC, folded alpha helix, very stable, 60 kilocalorie, minus 60 kilocalorie in delta G, and you have in other part amyloid assemblies. Or let's say all the plethora of assemblies that you can have, and these two can coexist. It's the question of multi-stability. The problem of alpha-synucleins or other amyloids that they are intrinsically disordered. So the assemblies or the system don't need. Okay, look, to induce the formation of amyloid from an IDP, you just have to template, to transmit an information of structures. In the case of PRP, you have to induce a unfolding first and a refolding. So you have two informations.
Yeah. Clear.
Yeah.
Yes.
It's why it's interesting. The system of PRPC is quite
like a system like beta-2 microglobulin somehow could be.
Yes, beta-2 microglobulin. If I'm right, but correct me if I'm wrong. There is a loop that's involved in the polymerization. That's hidden. Is hidden. I don't know if it's hidden or
This folded.
It's for that. Okay, so I didn't know.
So it is something that okay, quite different from prions. But
Okay.
if this notion holds true, probably it could be an interesting paradigm in this sense. Thank you very much.
You're welcome.
Thank you very much. There is no more questions, so we will thank again Sven Saupe and Rezaei for your both presentation.
Thank you.
And we will welcome you next week again for the next ZOOMinar. Thank you very much.
Thank you.
Bye-bye.
Bye-bye.
Bye.
Thank you very much.